Hello and welcome to another episode of Science on the Edge. In this particular podcast, I'm going to be talking about aging, which is kind of my favourite subject. In my master's degree, my final project related to aging, the effects of resveratrol, a compound that's found in grape skin, and its effects on DNA damage and repair. I also revisited this topic as part of my PhD, so it's a topic I'm very interested in. Aging affects us all, and most of us try not to think about it. We even put our old people, at least in the West, we put our old people into homes and sort of hide them away from society, like we're embarrassed by the process of aging. But maybe it's not that we're embarrassed. Maybe it's more that we just fear aging, which is understandable. I mean, aging is something to fear. Most of us do not have a pleasant experience towards the end of our lives. We get diseases such as Alzheimer's, Parkinson's, heart disease, arthritis, various other forms of dementia, liver failure, etc., osteoporosis. I mean, the list goes on. All of these diseases become more prevalent the older we get, and they are not fun to endure and do sooner or later result in death. So aging, by the way, don't worry, I'm not going to be spending the next hour talking about all the depressing aspects of aging. That will be part of it, but I intend to be a bit more positive. I'm going to talk about the process of aging, why we age. I'm then going to talk about what we can do to make the process a little more fun, to lower the chances of getting some of the detrimental diseases that are commonly associated with aging. And I'll also talk about the future of aging research, and it does seem to have a bright future. I mean, when all's said and done, our bodies are just machines. They're just biological machines. Regardless of your religious predispositions, if you have any, we are a machine. We repair this machine constantly. That's what doctors do. And as it's a machine that we still don't fully understand, there seems to be huge promise in the future for the development of new therapies, new drugs to fix this machine, to extend longevity. And by extend longevity, I don't mean to actually prolong suffering, to have people survive in old folks' homes for longer and longer with more and more pain and debilitating conditions. What I mean is, in the future, we should have the ability, and we're starting to develop the skill set for this now, to extend healthy life, to not just live longer, but to live healthier for longer. So, for example, I personally, this might sound very optimistic, but personally, I think that in the next few decades, you're going to look at a 100-year-old and he's going to be indistinguishable from a 30-year-old. That sounds like science fiction, but really the technologies that are being developed right now are going to turn this science fiction into science fact. We're already making huge progress in animal models, and it's only a matter of time before the breakthroughs with extending longevity in animal models actually make it into modern medicine, and we start living longer and healthier lives. Okay, so what exactly am I going to be talking about in this podcast? Well, first of all, I'm going to talk about what exactly aging is. I'll sort of give you some definitions. I'll explain the different disciplines in aging research. I will then talk about the different theories of aging. There are quite a few. In my opinion, no single theory is 100% correct. There's a lot of overlap. All the theories have certain merit to them. Once I've gone through the different theories of aging, I will then talk about current research into extending longevity. And then I'm going to let my imagination go a little bit wild, just a little bit, hopefully in a scientific way, by talking about the future therapies that should be available in just a few years. Hopefully before you and I get to the sort of age where we start suffering the negative consequences of aging. Okay, so let's begin. So first of all, aging. What is aging? Well, first of all, aging itself, it's a general process whereby the body changes over time. The field of aging involves the study of genetics, biochemistry, cell biology, endocrinology, comparative biology, demography, regenerative medicine, definitely. If you want to know a bit more about regenerative medicine, I really do recommend that you check out my podcast on stem cells. Aging-related diseases, that's one aspect of aging. Bioethics and evolution. Evolution is quite key. We are an evolved organism. We've evolved for a purpose. Well, purpose is probably stretching it, but the point is that our main reason for being is to pass on our genes to the next generation. That's really what started evolution in the first place. Now, we age, and different organisms age at different rates. Now, everything that you see around you in biology has an evolutionary reason behind it. And there's no reason to think that aging also has an evolutionary reason. Some animals age quicker than others, some age slower. Natural selection must play a role in that process. And I'm going to talk about that as well. Okay, so questions about aging. Well, first of all, if we look at comparative biology, we find very quickly by comparing different organisms that different species age at different rates. I mean, a mouse can live just a couple of years. Something like a chimpanzee can maybe live 50 or 60 years. Human can, in some cases, live to be 100, 110, 120. My memory may not be serving me correct, but if I do remember correctly, the person that holds a record for the longest life expectancy was a French woman who lived to be in her 120s. She actually used to sell paintbrushes to Van Gogh when she was young. Now, in other species, aging can vary massively. So, for example, the bristlecone pine can live up to 5,000 years. And the Dahlia anemone doesn't seem to age at all. Just seems to carry on and on and on and on. We're still working out why. There's also other species, such as the naked mole rat, fascinating but ugly species. Look up an image, the naked mole rat, really ugly creature. However, they don't seem to age. They really don't, and they don't get any of the diseases associated with aging. They don't seem to get cancer. They don't seem to get any neurodegenerative diseases. They just exist and keep on going. Eventually they do die, but they don't seem to age before dying. That's a fascinating creature, actually. I'm not going to talk about it much in this particular podcast, but I'll probably revisit it again. Okay, so the basics of aging. Let's get some definitions out of the way. First of all, let's compare aging versus senescence. Senescence is the decline of fitness components of an individual with increasing age owing to internal deterioration. Michael Rose came up with that definition, not me. Gerontology. It's the scientific study of the biological, psychological and sociological phenomena associated with old age and aging. Geriatrics, different from gerontology. This is the branch of medicine that deals with the diagnosis and treatment of diseases and problems specific to the aged. And then we have biogerontology, the study of the biology of aging and longevity. There's a few overlaps between these definitions, but I think providing these definitions at the beginning of the podcast will make the rest of the podcast flow a little bit better. Okay, so during this podcast, I'm going to discuss a few categories or aspects associated with aging. I'm going to talk about inflammation, I'm going to talk about telomeres. Don't worry if you don't know what a telomere is. I will discuss that later. I'm going to talk about free radicals. I'm going to talk about the wear and tear theory of aging. I'm going to discuss senescent cells, which increase in numbers as we age. And I'm going to talk about the evolutionary basis of aging. At which point I'll start talking about some discoveries and some therapies which seem to help with the process of aging. I'll discuss isothiocyanates, specifically sulforaphane. Again, I've already put a podcast out about isothiocyanates, specifically sulforaphane. I recommend listening to that. It's quite a short podcast. It's one of my bite-sized podcasts, but amazing compound, sulforaphane, that seems to retard some of the negative aspects of the aging process. And it is a compound that you can find in foods such as broccoli, but I'll get to that later. I'm then going to talk about fasting and calorie restriction and its effects on aging. This is quite an interesting area that's just opening up now in the last few years. We've known about calorie restriction and the fact that restricting the caloric intake of certain animals increases their longevity and their health. We've known about this since, I think, the 1920s or the 1930s. But in recent years, the science has evolved a little bit more. For example, time-restricted fasting seems to have quite a positive effect on health and longevity without the need to restrict the number of calories that you take in. But once more, I will get to that all in good time. Okay, so let's start with inflammation. Now, most diseases of old age have been linked to an increase in inflammation. That's not an exaggeration. So many diseases, which in the past were thought to have nothing to do with inflammation, seem on further inspection and investigation to be inflammation dependent. For example, Alzheimer's disease, Parkinson's disease, arthritis, and recently even autism has been associated with inflammation. Certain drugs which reduce the levels of inflammation have been tested on autistic patients, and some of the symptoms of the autism have been reduced. Now, this is an ongoing story, so if you do know someone with autism, don't think that all you need to do is bring down inflammation and your child's autism will be cured. These studies are in their infancy, but preliminary studies do seem to show that reducing inflammation has a positive effect on that condition. So, inflammation, how does it cause aging? What's its link with aging? Well, when it comes to inflammation, anything that causes inflammation in the body basically causes damage to the body. Inflammation involves the recruitment of certain immune cells to a particular part of the body. They are attracted to a region of the body, usually by some sort of damage to that organ or tissue. And damage to organs and tissues occurs throughout life. When these immune cells reach an area of damage, they cause inflammation. The purpose of that inflammation is to basically to repair the damage to that tissue, to that organ, to that system. So immune cells will remove damaged tissue from a region of the body, and they'll start the repair process. Unfortunately, there is collateral damage. Wherever there is inflammation in the body, you don't just lose healthy cells. You lose I will repeat that. You don't just lose damaged cells or dying cells. The process of inflammation also causes healthy cells to become damaged. And this can cause a bit of a cascade, and the end result over many, many years of repeated bouts of inflammation in various tissues and organs of the body is an accumulation of damage to those organs and tissues, which can result in all sorts of diseases, including Alzheimer's, including arthritis, including many other conditions. Now, part of how this works is inflammation itself So imagine there's some damage to, I don't know, some liver cells, for example. Now, this will result in the release of certain cytokines, chemical messengers, into the bloodstream. This will attract immune cells to the area. Now, these immune cells will clear up the damage, and in that process, perhaps damage some other cells in the tissues. Now, as cells become damaged, they can either die, they can basically go into apoptosis. Again, I have a podcast about cell death. It talks about apoptosis and necrosis and autophagy. So a cell can go into apoptosis, which means the cell dies in a controlled manner. It shrinks, it sort of digests itself, and the debris of that cell is just absorbed by neighboring cells. So a cell can go into apoptosis. Alternatively, a cell can become damaged and simply leave the cell cycle. What this basically means is it stops behaving like the type of cell it is. So if it's a liver cell, it stops behaving like a liver cell. If it's a kidney cell, it stops behaving like a kidney cell. And it kind of just sits there in the organ. Doing what, you might ask? Well, basically releasing very small numbers of inflammatory cytokines, chemical messengers that attract immune cells to the area and cause more damage. These senescent cells, therefore, build up over time. So the older you are, the more damage that has occurred to your organs and tissues, and as a result of that, the more senescent cells have accumulated in those organs and tissues. And these senescent cells attract more and more immune cells and cause more and more inflammation, more and more damage. So they accumulate throughout your life. These senescent cells don't just sit there releasing inflammatory cytokines, by the way, attracting immune cells, I should say. These senescent cells also fail to perform the normal actions of the type of cell they should be. So you end up with a liver that has accumulated all these non-liver cells that are unable to perform the functions of a liver cell, which means the organs slowly fail. They just cannot function normally. The same occurs in your kidneys, in your brain, all throughout your body as you age. So senescent cells in large numbers are bad. Now, if you're young, if you're a child, if you're in your 20s, you will have no senescent cells or very few senescent cells in your organs and tissues. So there's very little inflammation going on. Therefore, the organs and tissues behave the way they should. They're healthy, they can perform their normal functions. As you get older, the senescent cells start to accumulate, however. By the time you're in your 50s, you have a noticeable number of senescent cells throughout your body, which means your organs and tissues are starting to lose some functionality. And this just continues. Once you get into your 60s, the numbers are higher, and by the time you get into your 70s, you usually have so many senescent cells in your organs and tissues that you're at risk of certain organs and certain tissues failing, and certain diseases that result from inflammation. Remember, senescent cells are causing inflammation. Certain diseases associated with inflammation start to occur, and you age. Okay, so let's talk a bit more about causes of inflammation. So I've mentioned that when organs and tissues become damaged, which occurs all the time, you generate senescent cells, you generate inflammation, you attract immune cells. But there are things in our diet that increase the amount of inflammation. For example, hyperglycemia. So hyperglycemia, which is basically high blood sugar, activates the body's inflammatory defense mechanism, causing the release of a number of inflammatory mediators and cytokines, and can eventually lead to organ damage. As the only glucose-lowering hormone in the body, insulin, produced by the beta cells of the pancreas, not only alleviates the detrimental effects of hyperglycemia through its metabolic regulation, but also directly modulates inflammatory mediators and acts upon immune cells to increase the potency or effectiveness of the immune system. So hyperglycemia is pro-inflammatory. Too much sugar in your blood is pro-inflammatory, whereas insulin is anti-inflammatory. Now, the problem is that many people, particularly in the West, but also in the Middle East and throughout the developing world now, so many people in the world these days take too much sugar into their diet. We've been sort of taught to believe over the last few decades that fat is the bad thing. Fat is bad. The more fat you have in the diet, the less healthy you will be. And there's a tiny bit of truth to that. Being overweight is certainly bad for you. But really, we've been kind of looking at in the wrong direction by focusing on fat. Fat is bad. Fat is not bad. Not necessarily. Sugar is bad. Too much sugar certainly is bad. Sugar, you see, increases inflammation. And inflammation causes all sorts of diseases. Fat on its own does not increase inflammation. So if you're someone that's trying to lose weight and you're thinking, I'm just going to cut the fat out of my diet, don't. First of all, reduce your fat intake, sure, if you're taking in too much. But the sugar is really the villain in this piece. Too much sugar in your diet can cause inflammation, and this can result in all sorts of detrimental diseases, such as heart disease, atheroma, all sorts of things. You need to reduce the amount of sugar in your diet really to lower the amount of diseases associated with overeating. Plus, sugar stimulates insulin, and insulin is necessary for the absorption of fat into our adipose cells. So without insulin, you're not going to get all of the negative effects of taking in too much fat. It's not going to be quite as bad as if you take in the fat with the sugar, because of the effects on insulin. So obviously lowering sugar is a good thing. It certainly is, because it will not only reduce the amount of inflammation that you get in your body and the various diseases such as diabetes associated with inflammation, it will also help you regulate your fat metabolism that little bit better. Okay, I talk about this a little bit more actually in one of my podcasts, the one on nutrition. I talk about this at length. So I'm going to sort of stop myself from running away on this point. Check out that podcast if you want to know a little bit more. Okay, so hypoglycemia in summary is pro-inflammatory, whereas insulin is anti-inflammatory. Now, the more sugar you eat, the less sensitive you become to insulin. You're producing it so much that your cells lose their sensitivity, and therefore inflammation increases. So keep the sugar levels down. Now, inflammation can cause all sorts of diseases. I've mentioned some of them. One disease that it can cause is atheroma. Well, it's linked with atheroma. So atheroma is the buildup of various fatty plaques, sort of scar tissue almost, within the arteries, which can cause heart attack, stroke, all these sorts of things. Now, the buildup of fat in the arteries is an inflammatory process. It's an inflammatory process. So to reduce the likelihood of developing things such as heart disease and stroke, you need to reduce inflammation. This was actually noticed by Leonardo da Vinci centuries ago and then forgotten about. Leonardo da Vinci, one of my favourite historical characters. Well, characters, he actually existed. So character maybe is the wrong word. Historical figures. Leonardo da Vinci made a lot of scientific breakthroughs, most of which were forgotten because, there wasn't really a process for doing anything with his discoveries back then. That took the Industrial Revolution to get to that point. But one thing Leonardo da Vinci was very fond of doing was basically dissecting bodies to see how they worked. There's some fascinating drawings, if you look them up, that he made of during his dissections. But one thing he noticed when looking at the bodies of old people compared with young people is a buildup of fat in the arteries and a hardening of the arteries. He basically discovered atheromas. Didn't know what they were, but took a guess even back then that they were associated with a process of aging. So Leonardo da Vinci, genius, really, really was. Okay, so let's move on now from inflammation. Maybe we'll come back to a little bit later, but there's a lot to talk about in this podcast. I certainly won't cover every aspect of aging, but I'd like to at least touch on each main area. So we'll draw a line under inflammation for now. Okay, so let's talk a bit about telomeres now. Telomeres basically have been known to, or at least suspected to be involved in the process of aging for a number of years. There's been a lot of controversy into exactly what their involvement is. First of all, let's talk about what telomeres are. So, all of our cells contain a nucleus. Well, it's not strictly true. The red blood cells don't have a nucleus, but most of our cells contain a nucleus. And within the nucleus, there is DNA. DNA is coiled tightly into structures known as chromosomes. We have 2 pairs of 23 chromosomes, so we have 46 chromosomes each. Not everyone does. For certain genetic conditions where we have more or less, but most of us have a total of 46 chromosomes, 2 pairs of 23. Now, these chromosomes are coiled up DNA wrapped around little proteins known as histone proteins. The DNA carries the instructions to make various proteins and to regulate the various systems of our body, to grow, to help us repair, and to basically fight disease and just go about living. So our DNA is very important, and the DNA is stored in chromosomes. Now, at the end of those chromosomes, there are what's best described as small pads, known as telomeres. Now, imagine kind of like the eraser at the end of a pencil, except you have one on either end. So chromosomes have these telomeres at either end, and they're basically protective. They protect the DNA. The DNA itself, the chromosomes that is, they get shorter every time the DNA is replicated. Cells are dividing all the time throughout our life, at least most of our cells are, many of our cells are, and as the chromosomes divide, the daughter chromosomes are a little bit shorter. Now, this would be dangerous if we didn't have telomeres. As chromosomes get shorter, you are losing DNA, you're losing the ends of the chromosomes. And if the DNA at the ends of the chromosomes was DNA that coded for important things, such as making insulin, making collagen in our skin, etc., then we would lose the ability to produce those particular proteins. Luckily, we have telomeres. Telomeres get shorter. They're made of DNA. They get shorter as the chromosomes get shorter. But basically, it doesn't really matter because the telomeres don't code for genes, not as such. So they protect our cells. They protect our cells as they divide. And this is all very well and good. However, you've probably already identified this, but there is a problem with this process. And the problem is that sooner or later, if we live long enough, those telomeres are going to get very short. Too short, in fact. And if they get too short, we're going to start accruing damage to our actual chromosomes, to the genes within our chromosomes, which will cause those cells to basically lose their functionality. So telomeres get shorter as we get older, and this eventually results in a choice. The choice that a cell has to make when its telomeres get too short is to either die, go into apoptosis, as discussed earlier, or to become senescent, to basically leave the cell cycle, to stop dividing. If a cell stops dividing, then it basically won't be subject, or at least not as subject, to further damage to its DNA due to telomere shortening. And as a result, it's less likely to become cancerous, for example, or to start behaving abnormally due to a loss of vital genes. So becoming senescent is a good strategy for a cell. It sort of can no longer behave the way it should. It basically, if it's a liver cell, can't do all the things that a liver cell can do anymore. If it's a kidney cell, it can't do all the things that a kidney cell can do anymore. But by leaving the cell cycle, it's not going to produce daughter cells with even shorter telomeres and even more damage. So becoming senescent is a good strategy. So as we get older, telomeres get shorter, and we all start off with different length telomeres. So our genes determine the average length of our telomeres. So basically, you may have long telomeres because your parents had long telomeres or your grandparents had long telomeres. So a bigger pad at the end of your chromosome. A bigger pad basically means it's going to take longer for the cells to become damaged and to become senescent. So having long telomeres has been theorized to be linked with living longer, to greater longevity and a healthier aging process. Shorter telomeres basically has been associated in many studies with a quickening of the aging process. So shorter lifespan and more symptoms prematurely of aging. A good example of this would be in certain progerias. So progeria is a group of conditions whose symptoms are a accelerated aging. So you've probably seen pictures on the TV or on the internet of small children that seem to already be old, that already have gone bald, they have arthritis, they have dementia even, and all other diseases associated with aging. Those children are suffering from progerias, a group of diseases that causes premature aging. And some progerias are caused by being born with a particularly short telomere. Telomeres that are so short that they wear out at a very young age, before adulthood. So the patient basically gets the various diseases of old age at a much younger time point. So telomeres are important. So it's the pad at the end of a chromosomes. Gets shorter each time a cell goes through mitosis. Mitosis is the process of cellular division. We need to perform mitosis whenever tissues are damaged. If your skin, your heart, your liver, your intestine becomes damaged, as it will over time, you need to replace the damaged cells. So other cells in the body need to divide, resulting in daughter cells with shorter telomeres. So telomeres, what can we do about them? Well, you're born with a certain length, that's true. But telomeres have been repaired in some experimental animals using an enzyme known as telomerase. Now, telomerase is an enzyme that we can produce. It's coded for by a gene in our cells. We all have it. This enzyme repairs telomeres, makes them longer again. However, in most of our cells, telomerase isn't active. But it can be switched on, and extra copies of the telomerase gene can be added using genetic engineering into animal models and even into human models. I'll get to that later. And this extends the length of telomeres. Now, what happens when we extend the length of telomeres? Well, we've done this in animal models. We've done it in mice, for example. And the result is usually greater longevity. The animals live a little bit longer and they age slower. They don't seem to get quite as many of the degenerative conditions associated with aging. So repairing telomeres seems to be a very good idea for increasing health during the aging process and longevity generally. But would this work in humans? Now, that's a big question. And it's a question scientists have been asking for, well, since the 90s, really. But it's kind of hard to do the experiments. To perform genetic engineering on a human being, a healthy human being, in the hope of extending their telomeres and slowing down the aging process, it's not the sort of process, it's not the sort of treatment that gets approved by the various governing bodies, such as the FDA in the USA, for example. Which is when it gets quite interesting. It's where it gets quite interesting. I'll tell you about a particular scientist, a very controversial scientist now, known as Elizabeth Parrish. Now, she founded a company called BioViva. Now, the aim of BioViva was to basically repair telomeres in humans to try and extend longevity, something that is not legal to do in most Western countries. So Liz Parrish, who is, by the way, a proper scientist, and she has all the relevant qualifications. She's not a quack. She has a team of scientists working with her in the company. She decided to go out to the US, where she's from, I believe it was to Columbia, to perform a series of gene therapies These gene therapies, the aim of which were to, well, there's two gene therapies basically. One gene therapy was, the aim of it was to increase the length of telomeres in a particular patient. I'll get to who that patient was shortly. And another therapy involved regulating a myostatin inhibitor, which basically would result, in theory, in an increase in muscle density. After the age of 40, our muscles start to lose about 1% of their mass per year. Things can be done about it, such as exercise, but on average, that's what happens. So this therapy, its aim was to increase muscle mass. Okay, now let's get to the patient. The patient that this test was done on was actually Liz Parrish herself. Liz Parrish at the time was a perfectly healthy woman in her early 40s. She didn't want to try this therapy on a volunteer without trying it on herself first, so she self-experimented. She got a team of scientists together. They measured her telomeres, so they took a bunch of blood samples and tissue samples from her body, and they measured the current length of her telomeres. She then flew to Colombia to receive two experimental gene therapies. One was a myostatin inhibitor, a drug that is being tested as a treatment for muscle loss. The other was a telomerase gene therapy, the drug that BioViva claims has reversed a cell's biological age. So she had the therapy, having had her telomeres measured first of all, and then she waited. She waited for the gene therapy to take effect, and then her telomeres were measured again a couple of years later. So it was in 2015 that she had the original therapy. She was 44 at the time. And then independently, she had scientists measure the length of her telomeres a couple of years later. So this is controversial. BioViva claims that 6 months after the treatment, the telomeres in her white blood cells had lengthened by 9%. And it's now a couple of years since the original experiments were performed, since the gene therapy was performed, and they claim that the telomere length has increased further. So, in theory, it was a success, although, even if it was a success, she's one person. Now, you need more than one person to prove a scientific concept really works. You need more than one patient. So maybe it did work with her. Maybe her telomeres are longer, and maybe this will increase her longevity. Maybe it will. Only time will tell. But obviously, we need to verify this in multiple experiments with multiple patients. But brave woman performing this experiment on herself. I believe it's done her company quite a bit of good. But whether we can take these results seriously or not, only time will tell. I believe that over the next few years, more therapies will enter the public domain, whose aim is to increase telomere length, and maybe this will increase longevity. Although not everybody believes that telomere lengths are the key component of aging. Many people think that telomere length varies, it does vary, but this variation isn't necessarily associated with how quickly you age. There's some competing papers, some saying telomeres are key, some saying they're not. So more needs to be done. More studies need to be performed into the roles of telomeres in aging before we can really say that the way to increase longevity and slow down the aging process is by modifying telomere length, increasing telomere length. Time will tell. Okay, so that's telomeres. Now let's talk about free radicals. Free radicals are also known as reactive oxygen species. They're produced as a byproduct of respiration and metabolism, so basically being alive. The longer we live, the more free radicals we produce. We're pouring them out in our cells on a day-to-day basis. These reactive molecules are just byproducts of metabolism and respiration, and they react with our DNA and anything else that they bind to in our cells. When these free radicals bind to, say, DNA, they damage the DNA. And this damage builds up over time. So the longer you live, the more free radical damage you get. This damage to your DNA basically results eventually in genes becoming faulty. And if these genes become faulty, then they're unable to produce the various proteins that these genes code for. And that can result in aging, really, in damage to the cells, in damage to the organs, in gradual deterioration of the body. For many years, we've known that free radicals are involved in the process of aging, but the fact of the matter is that free radicals cause damage, yes, but when we're young, it doesn't matter so much because we seem to have a very good ability to repair that damage and to even mop up the free radicals as they're generated in our cells. That ability slowly gets lost over time, however. So free radicals, in my opinion, are more of a symptom of aging than a cause of aging. We all produce free radicals, but young people, healthy people, seem to be able to mop them up and deal with the damage that the free radicals cause. As we get older, we lose that ability. So free radicals are part of a story, but in my opinion, not the whole story. Free radicals actually became quite big when they were first discovered, especially once we found out that compounds known as antioxidants help to mop up free radicals. So antioxidants include vitamins such as vitamin C and vitamin E. And it was found many decades ago that if you take cells that you grow in culture, they can divide a certain number of times before they become senescent, before they stop dividing. It's known as the Hayflick limit. Cells can divide, then they stop as the damage accumulates to them and as the telomeres shorten. Now, if you take these cells in culture and you treat them with vitamin C or vitamin E, these are antioxidants. These antioxidants react with free radicals as they're generated, and they mop them up. And as a result, these cells grown in culture can divide many more times than they otherwise would. So you look at this experiment, these series of experiments, and you think, aha, we now have a way of extending longevity. Just take lots of vitamin C, lots of vitamin E, and you will mop up the free radicals and you will age slower. Unfortunately, it doesn't work. We've done the experiments. It might work in cell culture when you put these antioxidants onto the cells directly, but it doesn't work like that in the human body. If you take lots of vitamin C in, for example, as a big drink, it's water soluble, so you can take a pint that's got thousands of milligrams, a pint of water that's got thousands of milligrams of vitamin C in it, and just down the drink. It will get into your bloodstream, and it will go through your bloodstream, and then it will go into your kidneys, and then it will go into your bladder, and then you will pee it away. So taking antioxidants isn't a way to increase longevity as such. The antioxidants just don't make it into the cells. Any extra antioxidants that we don't need, they're just removed from the body. And in some cases, for example with vitamin E, they can actually be toxic to cells in certain parts of the body. For example, one study showed that vitamin E is toxic at high doses to the kidney, for example. So free radicals are important. Antioxidants do mop them away. Antioxidant supplementation, however, doesn't seem to increase longevity. Okay, anyway, moving on from free radicals, I may come back to them later. The wear and tear theory of aging. So organs, tissues and cells gradually build up damage over time, resulting in the symptoms and appearance of aging. That's basically the wear and tear theory of aging. This eventually results in disease and death. So the longer we live, the more damage occurs to our tissues and our organs. Wear and tear happens. Every time you bend your legs, you damage your knees a little bit. Eventually, you mess up your knees completely. That's, in summary, the wear and tear theory of aging. It's a bit more complicated than that, but the idea is simple. The older you get, the more wear and tear has occurred to your body. Eventually, this results in something failing. The heart failing, the liver failing, the kidney failing, the brain failing, and we die. The wear and tear theory is sound. It is sound, but as stated previously, it's not the whole story, because when you are young, you still get wear and tear. You get damage to your arms, to your legs, to your joints, to your heart, to your kidney, whenever you do anything in life. But when you're young, you can repair that damage, and you can repair it pretty much flawlessly. As you get old, you can't repair it the same way. So yes, we do suffer from wear and tear as we age, and that does result in many of the symptoms of aging. But when we're young, we can repair the wear and tear. And it's still not totally clear why, as we get older, we lose that ability. We lose the ability to repair our damage quite so effectively. This is an area that's under investigation in many labs around the world. Why is it that we stop being able to repair our bodies and our tissues effectively as we age? Okay, so moving on again. I've talked about senescent cells. I mentioned them in reference to inflammation. Now what I'm going to do is go into a little bit more depth. So now I'm going to talk about senescent cells. I've broached the subject briefly already, but senescent cells are so important, I think I need to go into a little bit more depth. So these are cells that have worn out or have become damaged beyond repair, perhaps due to telomere shortening. They can result from wear and tear, can result from gradual degradation of the telomere over time, so as we get older. One of the reasons that senescent cells accumulate, in fact. Now, these senescent cells, as they accumulate in our tissues, and they do start to accumulate by the time we get into our 50s, cause more and more inflammation by attracting cells of the immune system. And this inflammation in the organs and tissues builds up as the numbers of senescent cells increase, and eventually can result in damage to our tissues, to our organs, and then they lose function. They start to lose function. As you get older, organs start to fail, or at least they perform their normal functions less efficiently. And this is in large part due to the accumulation of these senescent cells. Now, if you've listened to a previous podcast that I did, which talked about apoptosis and cell death, you might be wondering, why don't these cells just die? Why don't they just go into apoptosis, controlled cell death, and then they'd be gone from the organs and tissues? We wouldn't get the inflammation, and the organs and tissues would survive and function better. Why don't they die? Well, there's a number of discussions that have been ongoing regarding why they accumulate. Now, this is rather complex, and I don't want to go into too much detail, but I'll give you a couple of the key points. So first of all, in a natural scenario, so by a natural scenario, I mean in a scenario where humans don't exist in cities, where we have everything that we want, all the food we need, all the comfort we need, in a natural setting, so we're talking the Paleolithic times, we wouldn't get the same number of senescent cells building up in our tissues. Why wouldn't we get them? Well, a number of reasons. The main reason is that you don't get to eat that often in the Paleolithic era. Some people do, many people don't. In the Paleolithic times, we didn't farm. We were hunter-gatherers. And in times of abundance, there would be plenty of food to eat, plenty of game to hunt, plenty of berries to harvest, etc., etc. And that would be great. But these times were usually punctuated by times of famine, times of scarcity, when there wasn't much food available. And when there's not much food available, our metabolism changes. Certain genes in our cells get switched on, other genes get switched off. I'm not going to go into specifics of which genes are switched on, which genes are switched off, but long story short, in times of famine, so prolonged fasting, we destroy our senescent cells. They're removed. They're not needed, so they are removed from our organs and tissues. And as they're destroyed, we also start destroying some healthy cells. We're basically just trying to make our bodies as efficient as possible so we can survive the time of famine until food is plentiful again, and then we can reproduce and pass our genes on to the next generation. So there's a selection pressure to survive these times of famine. So to survive these times of famine, we destroy senescent cells. We basically slow down the aging process. And as a result, our bodies endure long enough for us to get back to a time of plenty and to reproduce. So we remove senescent cells during times of famine. And in modern societies, we don't really have times of famine anymore. We have all the food we need. Well, to be honest, let's be honest, we have far more food than we need. Obesity, overindulgence is an issue in modern societies, not starvation, not famine. So, senescent cells would normally be removed, and in modern times they are just not. They accumulate over time. They accumulate over a lifetime. So by the time you get into your 50s, 60s, 70s, you have so many senescent cells that they can cause many of the diseases of aging. So going back to the Paleolithic times, we don't know this for sure, but it has been theorized that humans just did not suffer anywhere near as many of the detrimental diseases associated with aging in modern affluent societies. They aged gracefully. They stayed healthy into late old age. You might think, oh great, all the hippies are right. We need to reconnect with our Paleolithic ancestors and we're all going to live long, healthy lives. Well, there's a teensy bit of truth in that, but let's be honest, back in the Paleolithic times, you still tended to die relatively young on average. Some people made it to old age, but you got eaten by predators or you suffered from some infectious disease. Infectious diseases were everywhere, or you suffered some injury, and with no modern medicine, the injury wasn't repaired, and once again, you died. So, yes, in the Paleolithic times, some things were done right. You might say part of lifestyle did result in less of the diseases associated with aging in modern times, but we still died relatively young on average because of all the other diseases and accidents and predation that were prevalent back then. Okay, so that's senescent cells in a nutshell. Now what I'd like to talk about is the evolutionary theory of aging. I touched upon this in the introduction. I mentioned that we age and we are biological organisms, therefore evolution must be involved in some way, because evolution is involved in pretty much every part of biology. It's how we arose. Different creatures age at different rates. Some age quicker than others, some age slower than others. There seems to be a bit of a correlation between the size of an organism on average and how quickly it ages. So, for example, a mouse, which has a very fast metabolism, produces lots more free radicals, etc., etc., ages much quicker than a human, which is much larger, has a slow metabolism, produces less free radicals, etc., etc. If you draw a graph with the small animals at one end and the large animals at another, you will see that as the animals get larger, the animals seem to live longer on average, which led a lot of people to think it's all about metabolism. It's all about metabolic rate. Creatures with fast metabolisms age quicker than creatures with slow metabolisms. However, this just isn't true. There's exceptions which just completely blow this theory out of the water. For example, a rat is a rodent, and it lives something like 3 or 4 years. A squirrel is also a rodent, and it's about the same size as a rat, yet it can live 9 to 12 years. It does vary quite a bit. So their metabolic rates are both fast, they're both about the same size, they're both rodents, yet they age at different rates. So metabolism and body size isn't everything. Something else is involved in this process, and it turns out it's probably selection pressures and evolution. So suppose there are mutations that are beneficial in youth, but at the price of a higher rate of aging. more individuals will survive to express the early benefit than will survive to suffer the higher rate of aging. Mutations like this can therefore be incorporated by natural selection. So basically, aging evolves as a side effect of natural selection in favor of mutations that cause a benefit during youth. So what does this basically mean? It means that in the purpose of evolution, really, if you can say it's got a purpose, is to pass genes to the next generation. It's as simple as that. If a creature is able to pass genes to the next generation, then those genes will accumulate in a population. If a creature is unable to pass its genes to the next generation successfully or not in large numbers, then those genes will slowly be removed from a population. So if you have a gene or a set of genes which make you incredibly fertile when you're young, but make you prone to many of the diseases of aging as your life progresses, these genes actually will be selected for, because creatures do not live indefinitely due to predation and due to disease. So if you've got genes which basically will allow you to reproduce quickly and before you get eaten and before you catch a disease, those genes will be selected for. And the genes will be passed to the next generation and will accumulate. If you didn't have those genes, if you basically had genes which would allow you to live a very long life, or of centuries, of thousands of years even, but would give you a low ability to reproduce in your youth, then those genes wouldn't be selected for, because you would get eaten or you would get a disease in the evolutionary past before you could pass enough of those genes to the next generation. So basically, and this has been borne out by various studies, animals that have a high fidelity, a high ability to reproduce at an early age, those creatures tend to reproduce and pass their genes on to the next generation. Whereas animals that are unable to reproduce at a young age, they don't pass their genes on to the next generation quite so effectively. This is one of the reasons why animals that evolve in safe environments where there's low predation, such as island populations, tend to live longer. Look at the giant tortoises, for example, in the Galapagos Islands that live for centuries. Animals that don't suffer predation or disease, they've got more time to reproduce. So genes that allow them to live longer can accumulate in those populations, compared with animals that exist in environments where predation is high or disease is high. They don't have the time to wait to reproduce. Therefore, they need to get over-reproduction done very, very quickly. And the genes that are involved in allowing them to reproduce very quickly can produce negative side effects such as inflammation and result in premature aging. And this leads me back to the rat and the squirrel. The rats live in environments where there is more likelihood of getting disease and being preyed upon, and as a result they tend to age quicker. Natural selection has pushed them that way, whereas a squirrel has less of those problems to deal with and therefore lives longer. What I'd like to discuss finally in this podcast, I know I've kind of whizzed through the different areas and fields of aging very quickly, just touched upon each one. In future podcasts, I'll go into more detail. But to finish this podcast, I'd like to talk about aging therapies and the future of longevity research. So, aging therapies, the fountain of youth, is it real? Unfortunately not. There's no fountain of youth just yet. People have been fascinated by the mythical idea of the fountain of youth for so many centuries. We all want to be able to live longer and healthier lives, or at least most of us do. But there's nothing we can do to retard aging to a great degree right now, but there are some things we can do to help us to age more gracefully, to stave off some of the diseases associated with aging, and to increase average longevity. Not by a lot, but there are some things we can do, and research is improving over time. I think there'll be some major breakthroughs in the next few years. So first of all, calorie restriction. Calorie restriction has been known about for decades. If you take an animal such as a mouse and you feed it on a reduced number of calories, so a number of calories that is slightly less than it would normally need to survive, maintaining all the correct vitamins and minerals, then that animal will change its metabolism. Certain genes will be switched on, certain genes will be switched off. One of the key genes involved in this process is a group of genes known as the sirtuins, so SIRT1 for example. It's involved in metabolism and insulin regulation, longevity, all that kind of thing. I won't go into details about that right now. But by restricting the caloric intake of an animal such as a mouse, we can switch on a kind of lean burn mechanism within the cells, and these animals live longer. These mice live longer than control rodents, and they are healthier for longer, and they suffer far fewer of the debilitating diseases associated with longevity. The results of these experiments are so striking that a number of communities have formed that actually try to perform calorie restriction on themselves. There are websites you can go to. Some people have been calorie restricting their diet for decades now, and these people do seem to age quite gracefully. Obviously, we won't know for sure until these studies have been performed on numerous numbers of people over several decades. Humans age slowly, after all. But preliminary evidence seems to show that calorie restriction does have some benefits in humans. Maybe not quite as striking as in mice, but some benefits. The problem is, though, with calorie restriction, that you get hungry. You get very hungry. You would, wouldn't you? If you're restricting the amount of calories that you take in every day, you're going to be hungry every day. And many people might say, do you want to live an extra couple of decades but be hungry for those decades and the decades approaching those final couple of decades? I've tried calorie restriction myself. I've known about this since the 90s. I've given it a go myself. I didn't last long. I did not last long at all before the craving for a burger or a kebab or whatever became overwhelming. I don't know how these people do it. But that's one method to increase longevity, potentially, and to slow down the onset of many of the diseases associated with aging. But we've moved on a little bit from calorie restriction. Now is a sort of, well, there's a modified kind of calorie restriction, which maybe I should mention, where instead of restricting the number of calories that you take in permanently, you do it in a kind of off-on protocol. So, for example, maybe you take 1 week or a few days every few months where you do restrict your caloric intake dramatically. And then you go back to eating normally. And then you do it again a few months later, and again and again and again. People have studied this kind of time-restricted eating, time-restricted fasting, and it turns out that you do get many of the benefits of caloric restriction. The number of senescent cells that you get in your tissues reduces when you do this. And symptoms of things such as diabetes can be found to reduce, things such as high blood pressure, cholesterol levels in the blood, etc., etc. So there are definitely many benefits to this, and it's far more easy to endure a few days of not eating very much when you know you can have a nice big curry at the end of it. So that does work. That does work. There's another variation of this time-restricted fasting, which basically involves, instead of taking a few days off every few months, where you basically just reduce your caloric intake, you do it on a daily basis, and you just have as big a gap as you can between eating periods. So, for example, you could have one large meal, say at say lunchtime, so say 12, 1 o'clock, and then a large dinner, maybe at 5 o'clock, and then not eat again until lunchtime the next day. So you go a long period of time where you do kind of fast, a period of say 17 hours or so between eating, 12 to 17 hours. And it's been found that when you do this, when you have a big chunk of each 24-hour period where you are basically fasting, you get many of the health benefits. This kind of time-restricted fasting has been promoted quite extensively by a scientist by the name of Sachin Dananda Panda. He's written a few papers about this. He showed that time-restricted fasting attenuated metabolic diseases arising from a variety of obese diets, for example, and that benefits were proportional to the fasting duration. He also showed that protective effects were maintained even when time-restricted fasting was interrupted. So you can interrupt the process every now and then, and it can still basically result in many of the benefits. So he also showed that time-restricted fasting stabilized and reversed the progression of various metabolic diseases in mice with types of obesity and diabetes. And he established that clinically relevant parameters of time-restricted fasting for preventing and treating obesity and metabolic disorders such as type 2 diabetes and various liver conditions in those mice. So many of the studies that he performed, mainly on rodents, may be relevant for humans. We've not done all the follow-up studies yet, but it seems that just restricting the time period that you take in food in a 24-hour period may result in many health benefits and a reduction in many of the diseases associated with the aging process. And this, once again, is much easier than generalized caloric restriction. I should really talk about how caloric restriction works briefly, actually. We're still working out the details, but it seems that when you fast for a period of time, especially a longer period of time, so if you have a fast, for example, of a few days, you use up your collagen. Sorry, not collagen. You basically use up your glycogen stores. You store glycogen, sugar, in your liver and in your muscles, and you only have so much. You only have a few thousand calories that you store in your body. You need it for energy. If you don't eat for a few days, you use up that energy store, and your metabolic processes need to then switch to using primarily fat as an energy source. So you start burning your fat primarily. So certain genes get switched on, certain genes get switched off to allow this to occur. And when this does occur, you basically start destroying your senescent cells. It's a byproduct of the process. You destroy the senescent cells that are accumulated in your tissues. This allows your organs and tissues to function more efficiently. At the same time, you start to control your insulin levels a little bit better and your insulin sensitivity because you're not producing insulin all the time. And this helps deal with things like diabetes, for example. One other benefit of being on this kind of fast is that as well as destroying senescent cells, you kind of prime your stem cells. I'm not going to talk about stem cells in any detail now. I've got a podcast on that. I do recommend it. But stem cells are cells that help to repair your organs and tissues, to generate new cells to replace lost cells within those tissues. You have many stem cells in your body. Now, during a fasting period, you prime your stem cells. They don't become active, but they become ready. And when the fast ends, you've now removed your senescent cells, and when you start to eat again, your stem cells come into a kind of high activity state, and you produce new cells to replace the lost or damaged cells in your organs and tissues, to basically replace those senescent cells. And as a result, the organs and tissues function better. So by fasting and then eating normally again, You remove senescent cells and you produce new cells to replace them. Organs and tissues therefore function better, and many of the diseases associated with, for example, aging, can be ameliorated to a degree. So that's how calorie restriction works, which is one of the reasons actually why going on a total calorie restriction diet, one where you just live on reduced calories for the rest of your life, may not be quite as beneficial as this intermittent fasting. Because if you're on a permanent caloric restricted diet, yes, you may remove those senescent cells, but you're not stimulating your stem cells to replace them. So you're just getting some of the benefits. Okay, so that's a little bit about caloric restriction. Now what I'm briefly going to mention, very, very briefly, because again I have a podcast about this, is the benefits of sulforaphane. Sulforaphane is a compound that I've done an entire podcast about. It's a compound found in cruciferous vegetables. That's things such as broccoli and kale and sprouts, that kind of thing. This compound seems to have a very positive effect on on health and on the diseases associated with aging. So these cruciferous vegetables, they include within them a compound within their cells known as glucoraphanin. And they have a separate compound known as myrosinase. Now, these two compounds, they are kept apart within the cells of the cruciferous vegetables. And when the cruciferous vegetable is chewed, so for example by insects, the myrosinase and the glucoraphanin come together, they're brought together. The myrosinase is an enzyme that then activates the glucoraphanin and converts it into something called sulforaphane. Now, sulforaphane is basically the important compound here. Now, in the context of insects, which predate on these cruciferous vegetables, the sulforaphane basically it's kind of an irritant. It sort of burns a little bit. It's bitter and it burns, and it causes the insects to leave the plant and go somewhere else. However, in a human context or in an animal context, if we eat these cruciferous vegetables, the sulforaphane becomes ingested into our body, so it will end up in our bloodstream, and this sulforaphane then gets into our cells, and it activates an anti-inflammatory molecule, a protein in our cells known as NRF2. Now, NRF2 is a kind of master gene, master protein. So the NRF2 controls many other genes. It controls the activity of many other proteins as a result. Sulforaphane activates this NRF2, which brings down inflammation. Long story short, if you activate your NRF2, it's anti-inflammatory, it activates various other proteins, inhibits others, end result is you bring down inflammation. So if you eat lots of cruciferous vegetables, you will basically get lots of sulforaphane into your bloodstream, and it will activate NRF2, which will bring down inflammation. And as I've mentioned numerous times in this podcast, inflammation is bad. Inflammation reduces longevity and increases the level and the onset rate of the diseases associated with aging, things like Alzheimer's, diabetes, heart disease, etc., etc. So basically, by taking in large amounts of cruciferous vegetables, and numerous studies have shown this, we can reduce inflammation and reduce the incidence of a number of age-related diseases, and perhaps even increase longevity, although the debate is still out on that. Okay, so get your cruciferous vegetables, and if you want to know more about cruciferous vegetables and sulforaphane, then I suggest you listen to my podcast, which goes into really quite a lot of detail about it. Okay, now, next I want to mention omega-3. You've probably all heard of omega-3. It's a fat that you get in fish. Omega-3 also reduces inflammation. It's good for the brain. You need it for your neurons to form properly, so it's a very healthy thing to have anyway. But it also brings down inflammation. So if you have a high amount of omega-3 in your diet, whether it's from fish or fish oil, then you reduce inflammation. And a number of studies once again have shown that high amounts of omega-3 in the diet reduce the incidence of all sorts of diseases associated with aging and health, and also just generally bring down inflammation, which is a good thing. Okay, so that's just a brief overview of the current state of play with aging research and how aging works generally. I think I've covered the main points. I will go into more detail in future podcasts. So what's going to happen next? What does the future hold? Well, it's very positive. It's very, very positive. There's so much research going on right now into which genes are involved in the process of aging. What genes get switched on as we age? What genes get switched off as we age? We're mapping the genetic profiles, the profiles of which genes are active or inactive at different points in our lifetime. And by learning this information, we will hopefully in the future be able to develop therapies which help to kind of reset our genetics, let's say, or more accurately our epigenetics, so that we can kind of revert an older messenger RNA profile, one might say. That's the genes that are switched on and active. If we can sort of restore the profile to a kind of more youthful state, if we can do this, if we can come up with therapies that basically almost hit the factory reset button and get our genes to express themselves in the way that they did when they were young, even though we're getting older, then perhaps we can stave off a lot of the diseases associated with aging and maybe increase longevity. There's a lot of research going on right now, and it shows so much promise. We could increase telomere lengths perhaps using gene therapies, perhaps involving the stimulating the production of the enzyme telomerase to repair telomeres. That might increase longevity. Perhaps we can come up with better antioxidant therapies, ones that can get into the cells and help mop up the free radicals a little bit better, so that perhaps we could have less wear and tear as we age. There's a lot going on, basically. It's a really fascinating area of research. Time will tell which of these future therapies actually reach fruition, and if we do end up with effective treatments that retard the symptoms of aging or substantially increase longevity. But I'm quietly optimistic. I think that will happen. I think in a few decades we're going to get some really great breakthroughs which will just change the world. They really will. If we could increase longevity by a few decades and increase the health of the aged so that it more accurately reflects that of the young in our society. So if an 80-year-old could look, feel, and have the immune system of a 30-year-old, then we will increase the productivity of our population, and we will increase the happiness of our population. I think it's a really good thing. So watch this space. Okay, that's all for now. I hope you've enjoyed the podcast. I really do recommend catching up on some of the older podcasts that I've done that also relate to aging, such as the one on nutrition and the one on sulforaphane, not to mention the one on stem cells. But future podcasts will be released that also relate to aging. It's just such an important topic, and it's a topic I'm really fascinated by. Okay, thank you very much, and I look forward to talking to you again very soon. Thanks again for listening. Hope you enjoyed the episode. If you did find the episode interesting and would like to know a little bit more, there are a number of ways you can do that. First of all, you could follow me on Twitter. My handle is @Dr. Mark Darcy. That's just all lowercase, DRMARKDARCY. You can drop me an email. The address is info@scienceontheedge.com. or you could visit the website, scienceontheedge.com. The website is still under development, but new content is appearing all the time. Also, if you would like to support Science on the Edge and keep these podcasts under production, then you can make a small donation by visiting scienceontheedge.com and clicking on the donate button. Thank you very much and hope to see you again soon.